Welcome and thank you for joining me today. I will be presenting research that explores how internal pacemaker cell mechanisms contribute to irregular cardiac action potential firing. Let's get started. I'll start the presentation by giving a general review of cardiac pacemaker cells before discussing the goal and hypothesis of a study. The experimental techniques used to study the pacemaker function will then be explained, and we'll go over the main findings and picture analysis. I will conclude by summarizing the study's findings and going over its wider ramifications. Let's quickly review the fundamental idea of cardiac pacemaker activity before we examine the experimental investigation. Specialized cells in the central node rhythmic cells can produce spontaneous and action potentials and are the source of the heart's rhythm. Their ability to fire without outside stimulation makes them special. Rather, their rhythm is driven by two internal mechanisms, which is the calcium clock, which is based on an internal calcium cycling, and the membrane clock, which depends on ion channel behavior. Heart dysrhythmias may result from breaking down in the timing of electrical signals caused by instability in either of these systems. This provides context for the research I will be talking about. The spontaneous action potentials produced by the pacemaker cells in the central node are the main subject of this investigation. These cells have two internal oscillatory systems, the calcium clock, and also known as the Ca2 clock, which is driven by the cell's regular calcium cycling, and the membrane clock, also known as the M clock, which is driven by ion channel activity. And then without any outside stimuli, the researchers postulated this rhythmic irregular action potential firing could result from disruptions in either of these clock systems. The researchers used the well-known computer models of rabbit central node cells to test their theory. They were able to independently model the effects of ion channel activity disruptions and calcium cycling disruptions. Thanks to these models, to find out these modifications would impact action potential rhythm, they added stochastic variability to particular model elements, such as a ryanodine receptor, opening the L-type calcium 2 currents. Every change state was contrasted with a baseline model that depicted regular health pacemaker firing. The effects of adding variability to the calcium clock are depicted in this graph and picture. In particular, irregular calcium transits occur inside the cell as a result of increasing random variation in ryanodine receptor openings. Their graph here illustrates how these specific erratic calcium signals led to erratic spontaneous action potential intervals. It's crucial to say that there was no outside stressors or pacing changes. This rhythmic firing occurs solely internally. The idea that internal clock instability can cause cardiac arrhythmia directly is strongly supported by this. So the membrane clock, specifically the L-type calcium current, is the main subject of this specific graphs and figures you see, especially right here, the first one. Adding randomness to this current in the simulation caused the action potentials to fire erratically, exhibiting duration variability and skipped beats. In age or disease pacemaker cells, there was a ion channel behavior that became less consistent, and these effects mirrored experimental findings. When noise was introduced to the HCN channel, similar results were obtained, suggesting that When the membrane clock is destabilized, several of its components may contribute to its dysrhythmic firing. So uneven action potential firing dramatically increases when both clock systems were interfered at the same time. Compared to either system alone, the effects were even more severe, indicating that the two clocks typically coordinate closely to keep the heartbeat steady. One may compensate when the other becomes unpredictable. However, when both are unstable, the cell can no longer pace itself consistently, which results in serious arrhythmias. So at the end, this research shows that arrhythmic action potential firing can result from an stringent disruptions in either calcium or membrane clocks of pacemaker cells. The results highlight how crucial stable clock coupling is to heart rhythm. This has significant clinical ramifications for arrhythmias caused by things such as stress or aging in which terminal noise increases. Therefore, by focusing on processes that maintain calcium cycling or ion channel function, dysrhythmias may be prevented or treated at their central root cause. So here are my references for today's presentation. I hope you have a wonderful day.